what are some of the things that you would recommend to prevent, degenerative disease in general or Alzheimer's in particular?
So the big six are exercise, sleep, diet, community. So having more friends is better, lowering stress, and staying mentally active. And then, I would also add sauna to that.
Hi, everyone. Welcome to the Polling Principle, episode 3. And this week we have Dr. Christin Glorioso. Thank you so much for being here with me today. We're going to talk about all sorts of interesting things related to longevity. Thank you so much for uh, for agreeing to talk with me.
Yeah, thanks so much for having me.
Cool. So let me begin with maybe a little bit of an intro about yourself, right? So you're an MD, PhD. And yes, you've been working in doing research and working in longevity for a couple of decades already, total. And you're working now as the CEO and founder of NeuroAge, right? Which is a company in the longevity space that has signed an aging clock for the brain, right? If I understand correctly, and we'll get into a little bit of the detail of all that stuff. But I'd love to hear a little bit to begin with. Well, what is your kind of career trajectory? How you started getting interested into the space, and kind of what was if there was a turning point that made you decide to work in longevity, or like, what was the path that brought you here?
Yeah. So really, the path that brought me here is my own family history of Alzheimer's disease. So that's where I would start the story. So my grandmother suffered with Alzheimer's when I was a kid, and my grandfather took care of her in the home. Now, unfortunately, it's in the next generation. My aunt is currently suffering, and I found out that I have a very common risk allele for Alzheimer's disease. Yeah, the ApoE4 allele, one of them. And about 20% of people have that risk gene, and it puts you at about 3 times higher risk for Alzheimer's in the future. So it's a problem that's near and dear to my heart. And the biggest risk factor for Alzheimer's disease is aging. Uh, beyond beyond the genetics. So, young people don't get Alzheimer's, and the risk increases exponentially past age 70. So, I started off on a traditional path. You know, People ask you, how do you design new therapeutics or diagnostics for Alzheimer's disease? They'll tell you, go do an MD PhD program, bench to bedside research, study the disease. And then, at some point in the future, when you're a tenure track faculty member, 30 years later, you can start a company. And so I started off like that. So I completed my MD and PhD in neuroscience at University of Pittsburgh's joint program with Carnegie Mellon. And I was studying neuroscience there in human postmortem brain. So there were a couple of criteria I had when I started grad school. I thought a lot of the drugs were serendipitous and didn't work that well in the neuro space. So I want, and part of that is, bad animal models. So I wanted it to be human based and unbiased and relying on genomics. So that narrowed down the places that I could do a PhD in that field pretty dramatically. And so my PhD is one of the oldest and most established brain banks, human brain banks in the country, at University of Pittsburgh Psychiatry Department. And when I walked in the first day of graduate school, to who was to become my thesis advisor's lab, Etienne Sibyl, he showed me what I think was one of the very first aging clocks ever. So this was in 2005, almost a decade before the Horvath clocks came out. And I just thought it was the coolest thing I'd ever seen, and started working on it. And then, as I went through the rest of grad school and finished up med school, I was trying to decide whether I wanted to do a residency or not. I thought I was going to be a neurologist. But at that time, there really wasn't a whole lot we could do for people with Alzheimer's. And I was really passionate about what I was working on in the lab. So I made the very unusual decision to not do residency or even apply, and instead continue to work on my project as a postdoc at MIT. Famous aging lab there, the Grunty Lab. It's the lab that David Sinclair and Matt Cable and all these folks came out of. Lenny himself also has a company called Elysium that some people might be familiar with. And I continued working on the research I was doing as a graduate student. Um, and before I had a bit of a COVID journey where I was predictive modeling for COVID. I think a lot of us had a bit of a COVID journey.
Yeah, it's kind of
Before moving.
important for a few years. Yes.
Yeah, yeah. It's, and there's a lot of similarities between predictive modeling for COVID and predictive modeling for Alzheimer's disease. So I entered the X Prize. I created one of the best COVID predictive model for the US for predicting COVID cases and was working in the nonprofit space. I still am partially working in the nonprofit space before moving to San Francisco and joining UCSF as the chief strategist of an aging institute there. Then I met my co-founder and I started NeuroAge. And really what NeuroAge is aimed at is reversing brain aging in order to prevent dementia. And so we know that brain aging is about 40% lifestyle. So there's this huge actionable piece. And the first step towards improving lifestyle is really being able to track things. So we give people a way to track their brain aging
Definitely.
and personalized recommendations. Yeah.
I think you touched on a lot of things. So let's see if I remember all the questions. So you mentioned one thing that, of course, anybody that has been in longevity for a while would understand, but not so many people outside of longevity, which is the main factor for Alzheimer's is age, right? As people get older, they tend to develop that disease. Um, why would you say that is, right? Like, even if, maybe our understanding is not complete, right? But like I said, because the damage accumulates, is it because there are things that happen as people get older that kind of leads to, contracting those diseases and so on and so forth? Because there are, of course, other diseases that are not as related to aging, right? Like somebody can get a cold at, 5 years old and 25 years old and 75 years old, right? And, maybe the impact of the disease is not the same, right? But in Alzheimer's, like so many other diseases, it's related to the age of the person, right? So why would you say that is the case?
Yeah, so I think that, a unique hypothesis on why this is for Alzheimer's. And I fall into more of the reprogramming camp. So I believe that aging is a program, a transcriptional program that leads to neurodegenerative diseases. And when I say program, what I mean is that there are changes in the levels of RNA levels of genes that go up or down with age that create what's known as the hallmarks of aging. And in the brain, there's specific brain specific hallmarks. So, yeah, you see increasing DNA damage, you see mitochondrial dysfunction, you see changes in the connections between neurons, so loss of the synapses between neurons, inflammatory processes happening within glia. So these are some of the hallmarks, but underneath all of those hallmarks are changes in the levels of genes that's driving that. And it's really just the rate at which that happens that determines who will get a disease and who won't. So, Alzheimer's, if you live long enough, if you live to be 95, nearly 50-50 chance of having Alzheimer's. So, it's hard to think that that's not part of the normal aging process in some way, if it's that common. And I think that's also true for heart disease and some other very common age-related diseases, that there must be something fundamental to the aging process that's driving that.
Yep. So it's not just, so to say, random, right? Like cancer, for example, is maybe a little bit more random in the sense that some mutations will happen, and then the longer you live, the more the chance of having mutations, right? You say there's maybe a little bit of um, something that is happening in the way that our cells typically differentiate and progress as we get older beyond that set of random changes, right, or accumulation of damage, right? There's something intrinsic, I guess, in the development of Alzheimer's.
Yeah, so I think it's these changes in levels of genes. I think that's more important in the brain than in some other tissues. So, if you looked at blood cell aging. That's related to whether how many stem cells you have in your bone marrow. And so those stem cells run out because the telomeres get short after each division, and so you run out of stem cells. So there's other mechanisms in other tissues. But I think for the brain in particular, you have the same neurons you've had mostly the same neurons you've had since you were born. So it's the processes happening within those neurons that put you at risk.
Um,
Yeah.
Another thing that you mentioned that was very interesting was how you started with that dual degree of MD, PhD, and then and decided to skip the residency to do research. I guess it's interesting how maybe from outside of the drug discovery world, a lot of people think, well, the doctors and the hospitals are the ones that come up with new treatments or whatnot. But those two worlds, I guess, are a little bit differentiated, right? The people that work on the science that brings new drugs and the people that apply that knowledge in a hospital taking care of patients, right? So, like, how do you see those two things, and like, why did you say to go one instead of the other?
Yeah, I mean, I think, MD PhD, maybe this is going to be a controversial opinion, but I think is a little bit misguided for program. If I had to go back in time, I would do things differently. And the reason I say that is that, you know, people often it's 2 full-time jobs, really, doing research and seeing patients. So people almost always lean very heavily into one or the other at the end of the day. So either they're doing a tiny bit of research and seeing patients 95% of their time, or they see patients 1 week out of the year, and the rest of the time they're a scientist. And so it's pretty inefficient way to do things. That's not to say that there aren't some people that are doing 50-50, and there's plenty of MD PhDs that have brought a lot of really great medicines into the world, as well as basic science. Um, but for me, the best way to bring something to people and to patients is just to do it yourself and start a company. So to me, that seemed like the most efficient route to actually get something out in the world. And academia, you have for the most part, you have one little sentence at the end of a paper that says this could be a drug someday, and then no one pursues it. No one knows how to pursue making that happen. Um,
Yeah, yeah. To be honest, this was one of the things that I always thought was very frustrating, right? Like, I remember watching people talk about longevity, 15, almost 20 years ago, and then you think, well, people have been doing all these experiments on mice, and worms, and flies, and even in mice, right? In mammals, we know how to extend the lifespan of those animals significantly through many, many, many different ways. But nobody, at least nobody yet, has managed to apply those and prove that they work in human, right? So it's kind of frustrating that we understand, quite a few options for how to increase lifespan, but it's becoming, really, really hard to translate to that into therapies that we can give to people, right? That we can take ourselves.
yeah. Well, I mean, I think in mice, right, the um interventions that have worked the best are caloric restriction, which is hard for people to do. Um, there's the dwarf mice, so they're low on growth hormone, which isn't a great route for people, unless you want to suffer in that way. There's rapamycin, which I think that's a whole other topic that extends lifespan consistently in mice. Um, then I think there's just mice aren't people, and there's a lot of things that just haven't translated well. There are some winners. PI3 kinase is in cancer drug, and that's age one in animals. That's one of the very first longevity chains that was discovered. Actually, is a medicine in people. And interestingly, the GLP-1 um, drugs, receptor agonists, do have some evidence to be going through PI3 kinase, so it going through longevity pathways themselves. So I think they're the first longevity drug. I think it's already on the market. It's my opinion. Um,
Yeah. But I mean, they have other issues, right? Like both density and not everybody can tolerate it, and so on and so forth. Right?
sure, they're not perfect. For sure.
Yeah. Um, So, I wonder also, um, I think you already answered this before, right? About why the brain specifically, as opposed to other, effects of aging or other, systemic effects. But I guess in some ways, the brain is the most important organ, right? Like, so, even if we lose a kidney, right? Like, I mean, there's always an option for a transplant, right? Or the liver, the liver, I guess, will regenerate over time, right? But if we lose brain function, there's no way to recover from that, right? Like, there's just not much more that we can do, right? Even if the individual would otherwise stay alive.
Yeah, that's a huge reason why I'm interested. It's very hard to replace the brain. We're gonna need um bigger guns to do that. And also, for me at least, if I end up frail in some other way and, maybe disabled or whatever, as long as my brain is still working well, I think my quality of life will be pretty good. So that once you lose that function, at least for those of us that live their lives in the world of ideas, it becomes really challenging. So,
Yeah, yeah. I mean, I personally believe it has to be a combination of things. I don't think there's any single thing that we can act on. Like, even if we somehow could prevent our brain from aging, right? Like, if sarcopenia, for example, keeps developing, one day we'll, fall in the shower and that's it, right? I mean, it's another very common reason why people, eventually, die early, right? Like, they have some kind of injury because they are weak, and then, once they're in a wheelchair, they don't exercise, and everything else goes down, right? They develop diabetes or whatever, right? And it kind of everything goes down the drain. So, I mean, I personally believe even if we could prevent aging for the brain or for any other organ, right? Like, just by itself wouldn't be enough to significantly push the needle, right? Like, I unfortunately we have to solve everything at once, right?
mhm.
Um,
Yep.
so let's talk a little bit about aging clocks, right? Like, uh, why are aging clocks important for people that work on longevity?
So, well, we need some way to measure the rate, some objective way to measure the rate at which people are aging so that we know which interventions are working in people. Because lifespan as a measurement tool in a clinical trial, that's a very long trial. We might not live to see the results of it. So you need some other way to see how fast people are aging. Um, so that's the primary goal, and then it also helps people track their own lifestyle. So, if I exercise more, am I slowing down my aging? As a whole, if I eat this diet, am I slowing down my aging? So, it's also good from that standpoint.
Yeah, yeah. So if I can maybe paraphrase it, the idea would be to find some kind of indication of how aged or how old an individual is that is not just the age, so that 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 marker, right, is so strongly correlated that we can use it instead of age as a endpoint in a clinical trial, right? There's like trying to explain it for people that are not necessarily familiar with drug discovery, right? But so we can use it as a metric of whether a new molecule or a new drug is useful for a specific purpose, so that when we want to run a trial for this, we don't have to um, do a 75-year experiment, right? But we can do an experiment over the course of 4 or 5 years. And then ideally, that experiment would say, we apply some intervention to a group of people, and then we give a placebo, for example, to another group of people. In the placebo, we see that the marker goes up with age. In the group with the intervention, we see that the marker goes in the opposite direction. And then we have strong enough proof that is working to prevent that aging, so that we can go to the FDA and say, hey, this is working in this specific way, let us give this drug to people, right? Like, that's the main idea, I guess.
Yep. Yeah, exactly. And the simplest aging clock is actually one that we all use every day, which is just, you know, how old does someone look? And so there is a clock that people have created called face age. And, that's some our own internal algorithms for determining that, but we're pretty good at telling how old people are just by looking at them, maybe plus or minus. 5 to 10 years, most people can tell. And you're not conscious of what is going into your own algorithm, but that's, wrinkles and posture and, gray hair or whatever it is. It's some combination of things that is pretty accurate. So, you could use that as a clinical trial endpoint. Some people might be doing that, but there's a lot of debate in the field over what the best clock is and what it should be measuring. Um,
Yeah.
so
Do you think that, um, I mean, I don't know about, the drug that you're working on, but that are, like I said, there are many, many other drugs. Do you think that if people do interventions like doing more exercise, changing their diet, would that significantly change any of these clocks, right? One of the things, and the reason I'm asking is, one of the things that I always found very curious is that elite athletes don't necessarily live longer than most people. As a matter of fact, many of them, don't necessarily, live, much longer, right? There's maybe a little bit of a curve, right, where the peak in terms of longevity is not necessarily at the end, right? So you have people who are incredibly fit for their whole lives, and then at 79, or whatever, right? Like they have a heart attack and they die, right? Or something else, right? So what is the I guess two questions, right? Like, what is the rationale for why maybe some of these aging clocks, don't capture some of that, fitness, and then, like, could people actually see the impact of some of these interventions if they use the aging clocks?
yeah, so I think the studies are just now coming out, but some of the aging clocks certainly do respond to exercise, to caloric restriction. I think there's been some papers on some. I think maybe it was metformin recently that responded. The aging clocks responded to. So we are seeing that, but different clocks respond to different interventions, and different clocks predict different diseases more or less well. So part of my definition of what an aging clock should do is predict multiple different age-related diseases. So it should be capturing the underlying biology behind, cancer and heart disease and neuro and all of these things. So that's kind of another way to look at it. But I think the first generation clocks by Horvath and colleagues are not moving very well with people's interventions, and that's because they're a little too overfitted. So they have really great correlations with chronological age, but they're missing that noise component that's actually the biological aging. So they have R squareds of like 0.99 with chronological age, but that means they're not moving with anything else. Um, so it's kind of a balancing act to design these.
Mhm. Nick. Um, Maybe switching topics a little bit. How hopeful are you that we will have uh, approved drugs for longevity, say, or a drug that may already be in the market that can be prescribed to people and is widely adopted, right? So not just a few biohackers and so on, but that the general public can just go and start taking for the purpose of extending lifespan. Do you think this is something that we'll see in you know, 5 years, 10 years, longer, right? Why, and I'll go a little bit deeper into why I'm asking that later. But, I'd love to hear, what you think about, how soon we'll say some of these things.
Yeah, I mean, so I already think there is one. These GLP-1 RA drugs. I think that those actually are longevity drugs. We see them working with multiple age-related diseases. They appear to be, have effects beyond just for weight loss and diabetes prevention, but additional health span and lifespan promoting effects through longevity pathways. So I do think they're already here. Um, we need what would be great is if we ran 12 aging clocks, the GLP-1 RA study, and showed that it moved, most of them. Then we would have maybe the evidence starting to accumulate that we need to really show to demonstrate that. I think we need to add these clocks to everything. It's not that expensive, so it would be great to kind of have that comparison across clinical trials. I thought the BioAge drug was going to be another longevity drug that was gonna hit the market. Unfortunately, they had, some liver toxicity in the clinical trial, but I thought we were gonna get there in the next couple of years with them. Um, and there's a lot of other companies that are working on things. So I do think it's gonna be in the next 5 years, but, you know, these, therapeutics companies are need to come out and show us all the evidence. They're gonna need to say, "Look, here's the mouse data extending lifespan in mice with this drug, and here are the human aging clocks, and here's the multiple disease indications." And then we'll start to believe that this is truly a longevity drug, I think, when we see all that evidence.
But I mean, just so the GLP-1 inhibitors, I think those are interesting, right? Like, in from what I understand, it's not that hard to get a prescription for them. I think there's even online, pharmacists or whatever. So it seems like within the realm of what a biohacker or somebody doing some self experimentation could do with relatively low budget, right? Just go get a prescription for it, use it for 6 months, do a few of these tests before and after, and you're talking about maybe a few $1,000s, right? Like single digit
Mm.
$1,000.
Mhm.
Um,
Yep.
so yeah, I would not advocate for somebody to do self-experimentation on a podcast, right? And but yeah, I mean, I guess maybe within the next, few years, we could see people actually, showing that. I mean, some people are already doing that with rapamycin, I guess, right? And that's how, I guess, some of the data was obtained. Somebody did a study, I think, a couple of years ago. Uh, I think it was in Matt Kaplan's lab, right? People that were casually using, well, not casually, but using rapamycin for longevity, right? And they saw some data that seemed positive. But I don't think I don't know. I haven't heard about it since it came out, probably a couple of years ago now.
Mhm. Yeah, and I think Brad Stanfield is running the rapamycin and, exercise trial. I think there's some aging clocks with that, so probably we'll get some information with that, and I think the dog aging project is looking in dogs. So, I think there'll be some more rapamycin data coming out, coming down the pike, but we'll see.
Yeah. Um, the reason I was asking this, right, is I think especially today we're seeing so much and so fast progress in other areas, right? Like, AI, I mean, I think is the most obvious one, right? But yeah, I don't think it's the only one. Robotics, for example, right? Like, people seem to be able to do things that even just 2, 3 years ago were science fiction, literally. And um, that's not the case in drug discovery, right? The drug discovery seems to, take quite a bit longer, right? And I mean, I would say life sciences in general, right? And all of medicine, progress just seems to be slower, right? So, I mean, I'd love to hear your take on why that is the case, right? And maybe what are some of the things that we could do to improve the rate of progress?
Yeah, so I mean, I think the biggest one is it's just very expensive, so you don't get very many shots. Even a medium-sized pharma company maybe has one or two shots on goal with a drug in humans. It costs $2 billion to go through all the phases of clinical trials. That's a huge stumbling block, right? What if we could just test all the drugs in people immediately? That would accelerate things dramatically. And then recruiting for clinical trials can be slow. The requirement for mouse data, running mouse experiments takes a couple of years before you get to the clinical trials. So there's just all these bottlenecks. AI is going to accelerate that. And I think then it's another reason to have all these aging clocks piggybacked onto existing drugs because I'm sure there are drugs out there that are affecting longevity that we just don't know about because 'cause we're not looking at it. Yeah.
Yeah. Um, do you think there is room for, some uh I don't know, magical model, machine learning thing that will come up one day and would be like, AlphaFold, but for, longevity drugs or something that, a model that, you feed it, the gene of a person and it tells you, what kind of molecule would actually help slow aging for that person. Would that even make sense?
Yeah, so there's something that I've been thinking about for a while, and I actually heard that there's I think a professor at Harvard now that's actually working on this. I was hearing a talk he gave at Biomarkers of Aging, and it is a virtual cell. So people have started working on the virtual cell. So if you can model what happens when you give a drug to every process in a cell. So, what happens to RNA levels? What happens to protein levels? What happens in a neuron to firing rate? If you, what happens to epigenetics? If you can put that into a computer and model all of that, it's very complicated. The compute power, it would be extremely intensive. We don't even have the brain mapped out in people yet. We don't know all the connections. We don't know the wiring diagram, right? So, once we have all of that, then you can just run virtual experiments. So instead of having to treat a mouse for 2 years or a person for 2 years, you can just simulate it. Here, we'll put this drug in, and we'll put this combination of drugs in. What happens? And I think we're going to get there, but we need to map it all first, and we need very powerful computers to be able to simulate all of that.
Yeah, I think I was actually looking it up right now. I think it was Martina Sitnik, Professor Martina Sitnik, and her group. Like, I think that was at least one of the groups that was doing this research on virtual cells. And I know there was another group sponsored by the Chan Zuckerberg Initiative. It's a nonprofit that you know that they have that was doing some research, but I don't remember the name of the other group. Yeah, I'm a little I think the compute capacity that we would need to simulate an entire set is a little bit too much for what we have right now. We don't really
Right.
know quite well how to do that, but within a few years, maybe. Yeah, I mean, it's very very hard to say, what would possible or not. Um, one thing that I'm actually very interested in is you mentioned um, mice studies, for example. I know there's another startup that is working on robotic automation of mice studies. So I think that would be interesting. I think maybe part of the reason why these things are expensive is handling animals and like moving things around. Uh, all the work that is involved in a lab, right? Extracting data, analyzing the data, copying the data back and forth, and maybe a combination of robotics and smart automation could help that. Right? Like the same way that we don't build things by hand anymore. Right? Like factories are pretty much fully automated end to end these days.
Yeah, I think that could definitely be helpful. But you still have to wait for the mice to age. So that's the time. So you're waiting for their biology, you're waiting for them to get their disease to progress, you're waiting for them to uh, live their life and die of old age so you can do the lifespan studies. So there's this biology limit. And mice are even bad, especially for neuro, mice are bad models. So you really want to do it in non human primates, and those studies take
Mhm.
even longer. Um,
Yeah,
so it's
like 15, 20 years. I think this is what a monkey, for example, would normally live, right?
At least, yeah.
Mhm. Yeah. Um, I don't think that's a good solution for, if you need to do a study waiting for the animal to age, there's just no way around that, right? Uh, it would be great if we had this validated clock that we could go and say, yep, we see the clock, go down, we run the experiment for 3 months and then we decide, yep, this is good enough. Let's move on and test it in humans.
Yeah, and I think we're gonna get there. And then the other thing is, all the organ on a chip stuff. So, organoids. So FDA said that they're gonna start accepting that instead of mice. So if you can, I don't know, grow a mini brain on a chip, and test it on, a mini human brain, then you start to get other answers. But that also needs to be validated. So I think we're still dicey how well that's gonna translate. But
Yep, yep. So maybe a little bit like to add a little bit of context, right? So because these organoids are like small samples of tissue, right, that are obtained from human cells that are typically modified a little bit, right? So they don't, die right away. And they are put in a shape in which they resemble an organ or a series of organs. And then they would be used to test mostly I guess toxicity, right? So you insert the drug and then look at it and say, well, does it cause the tissue to die or not, right? And I guess if you had a model that was good enough, for example, in the case of cancer, you could also test efficacy. You could look at it and say, does it kill the cancer and leave the tissue alive, right? And then in
Mhm.
that case, think, well, this may be a good drug for cancer. My understanding is that it's very hard to create these things as models for other diseases, right? Like you can't just, create a, you know, organoid model of Alzheimer's and then, look at it and say, well, that's it, does it cure Alzheimer's in this little, neuron tissue or not, right?
Yeah, I mean, there's companies that are doing that and academic labs that are doing that, and they even have them, some of them have quacks and tangles. You can do experiments where you look at how the neurons fire and wire together. So, it's new, I'd say this is the last 5 years that people are creating these things and they're getting better over time, but that's definitely where everything is moving.
Yeah. Um, and then I guess the main benefit is that time would be reduced and then the cost would also be lower, right? Like I don't know how expensive are the organoids. I know a mouse would be a few $100s, right? Maybe a few thousand if you need to take care of it for many years on average, right? But I guess these organoids would be a little bit more expensive because they are newer. I don't know. I don't have a good intuition for how expensive it would be to test them on them.
I think at this point they're a little bit less expensive than mice. So you could do like you could do experiments in 6 mice, which is like a very minimum for a couple hundred grand, is where we're at. And the organoids maybe half that kind of ballpark, but the- over time. Yeah.
Yeah. Yeah. I was going to say because you would need several dozens of them, right?
Yeah.
For
Uh, yeah. You're talking about for
No, I mean for mice. Yeah, in mice, you would need many dozens of them because there's variability. I guess for the organoids, even you could use a lot less, right? Because if they are industrially manufactured, the variability should be small.
Right. I think at this point, they're very much not industrial manufactured. People are doing this by hand. I think there is some variability, but eventually that's where we'll be, and it'll be cheaper. Yeah.
Mm. Um, I think the one thing that would be interesting, right, is what else can be done to lower the cost on timeline. I think so many people keep mentioning how sequencing, for example, benefited from Moore's law, right, and the first genome was sequenced for $2 billion, $3 billion, and now you can get a whole genome sequencing for literally $400, right? Um, Everything, the entire genome, intron sections, everything, right, and I wonder why that hasn't translated to other areas within life science, right? Like if people had managed to apply that same cost reduction principle to other things, recruiting patients for a clinical trial, right? Testing, toxicity on all sorts of other organisms, right? Like maybe it would be much easier to come up with new drugs today, right? But I don't know why there's not enough impetus, interest in trying to drive down those economics.
Yeah, I'm not sure. I mean, I do think we need maybe help from the tech industry and patient recruitment. That's pretty fertile ground for getting those clinical trials recruited and off the ground quicker. Um, that's part of, at NeuroAge, that's part of the thinking is once we have enough people that have taken the NeuroAge test, then we can easily, for people that lifestyle alone is not enough to prevent them from getting an age-related neuro disorder, we could funnel them to a clinical trial that might be helpful for them, based on their biomarkers. But that's part of what we're trying to do. But I think there needs to be a lot more of that. We need the big tech companies to come in and help us solve that problem in the way saying, Google solved Google Maps. We need this for clinical trials, right?
Yeah.
So,
Yeah. I mean, it would be terrific, right? I personally I don't understand why not enough people are interested in this type of things. But yeah, I don't wanna rant either. So maybe going down a little bit on that, right? So you mentioned lifestyle, you mentioned interventions for neurodegenerative disease, I guess, not just Alzheimer's, right? But maybe what are some of the things that you would recommend people in general to do to prevent neurodegenerative disease in general or Alzheimer's in particular, right? Like, what kinds of things people could do in the day to day, right? Over many years.
Yeah. So the big six are exercise, sleep, diet, community. So having more friends is better, lowering stress, and staying mentally active. So those are the big six ways. And then, I would also add sauna to that. What you do that's good for your heart is also good for your brain because um a lot of what happens with vascular dementia and strokes and everything else are just cardiovascular problems in your brain in those blood vessels. So, statins are good. The GLP-1 drugs have some evidence. In women, there's emerging evidence for hormone replacement therapy. Alzheimer's is about twice as common in women as men, and part of that may be due to menopause and lack of female hormones in post a certain age. So those are some of the big ways. Another one that I often talk about is nicotine. So nicotine itself is a nootropic. So it's actually very good for your memory.
Yeah.
Um, but the cancer causing part of that is due to the delivery system. So the smoke or the vapor that gets in your lungs and causes cancer. But nicotine itself is actually a great drug and it's in phase 2 clinical trials right now. So I think we may see people on a nicotine patch or a gum or a mint or something in the future. And people always talk about Jean Calment, who's the oldest woman ever to live. She lived to be 122, and she took up smoking in her, you know, after her 100th birthday. She started smoking, and I don't think that's an accident. So we see less Parkinson's in people that smoke, and I think it's if you can survive the heart disease and the cancer, get enough genes to do that, then it's actually protecting your brain. So that's one that I think
Oh,
will
interesting.
have some attention.
Yeah, I
Yeah,
mean, I wanna be careful also, because I don't know who may be listening to this, but I guess, if somebody picks up like these, zinc patches, right, or like nicotine patches, that may not be necessarily as bad as start smoking and so on, right? And I guess also, if you don't necessarily smoke, right, like there's no risk of lung cancer, right, which is the main.
right.
That's the one.
Yeah, that's when you don't start smoking, that's bad for you, for sure. The risks outweigh the benefits there, but, you know, I was in my 80s and starting to have some memory problems. Would I have a nicotine patch? Probably. Yeah, that might make sense.
Yeah. Um, Actually, I wanted to ask another thing about the sauna, but before that, do you actually take any nootropics apart from? I mean, I don't know if you take any of them, right? But we talked a little bit about nicotine. Nicotine could be considered, I guess, a nootropic, right? But there's many others that people have taken over the years, right? Ginseng or like ashwagandha or whatever, right? Like, do you take any of those, or would you recommend people take any of those? Rhodiola, I think, is one that I take that has worked very well for me. But yeah, I mean, I'd love to hear your take.
Yeah, so what do I do? Um, I actually do HRT. I'm, perimenopausal. I started that about 6 months ago. And I take a statin. Because of my ApoE4 allele, that also puts me at higher risk for high lipids and high cholesterol. I don't have the other risk factors like diabetes or heart disease or any of that. So my doctor was like, maybe you don't need a statin. But because of the Alzheimer's risk and because of my, high lipids that are genetic, I do take a statin. There's a lot of nuance with that. I try to do sauna. Sauna I do mostly with hot yoga. So I think hot yoga is like the best thing ever because it hits ticks a bunch of boxes at once. So you get the salt release that's good for your blood pressure of sauna and the meditation. And then if you go to a hard enough class, you're also getting your heart rate up. And there's push ups and sit ups, so you get some resistance training as well. So I do that. Um, but I don't take ginseng or ashwagandha or any of those. I honestly haven't really looked into it that much. That's probably reading I need to do on those. I do drink a lot of coffee. So I think caffeine is a bit of a nootropic.
Yeah, I guess it's maybe good for the brain, and I also, I think if I remember right, there's a little bit of a vasoconstrictor, right? So it helps when you wake up with a headache or whatever, right? But,
Mhm.
yeah, maybe not so great for the stress, that you mentioned too. Um,
Yeah, but it's actually, it has good evidence for lifespan, and it's not the caffeine actually in the coffee and it's coffee and tea, it's the polyphenols, so, the antioxidants in the beans that are good for people and the tea leaves.
for sauna, I guess maybe one other good thing about it is the fact that it makes you sweat, right? Then I don't know. So many people have been talking about microplastics accumulating and this and that. Like, do you think there's any relevance to that? Like, maybe there's toxins or other things that we kind of release when we go to the sauna or do other activities that help kind of cleanse.
Yeah, I don't know if you can get rid of microplastics from sweating. That I haven't looked into, but I can tell you there was a Nature Medicine paper that came out about a month ago where they showed, a couple of teaspoons of plastics in people's brains. They actually could see them under the microscope. And that's the plastic, really common plastic. So I was scared of the BPA, which is, but apparently that doesn't accumulate in your body. That's like a proestrogen or something is the reason people avoid that one. But this is PE, which is in uh polyethylene, which is in like plastic bags and plastic bottles and things like that. And so that is what's actually accumulating in people's brains
Yeah.
and
And
other organs.
receipts, right? Like the little paper that they use for like the credit card receipts and all that. Yeah.
Yeah, and the nonstick coating on pans. So I went and got rid of all of my pans with nonstick coating and all of my Tupperware that was plastic because it's worse if it gets hot. So if you microwave things in plastic containers, that's really bad. And all of the plastic bags, I replaced that all with glass and replaced my pans with stainless steel or ceramic and all the plastic ice cube trays and spatulas and stuff with silicone. So we don't know whether it was causal, but it's scary enough looking to me, and I feel like the downside is not that much to doing, a couple $100 you can revamp your kitchen,
Yep.
and
I mean, people say,
yeah.
correlation is not causation. Yeah, but it's still correlation, right? So there's still, it's still probably worth looking into. Yeah, I remember getting rid of all my plastic cutting boards, a long time ago as well. And Teflon, I think that the Teflon, pans, I guess, they're not great either. Um,
Yep.
maybe one more thing about the sauna, right? I wonder if, I guess like when you go through the temperature transition, right? Like that is good for the cardiovascular system. How about cold therapy? How about a cold plunge? Or I think it's just this Wim Hof method, right? Where people kind of go through like extreme cold for a few minutes and then take a break or whatever. Like do you think it has the same effect, better or worse?
Yeah, I did do a deep dive on the cold plunge as well because I live in San Francisco and I live near the bay and there's some folks that go swimming in it. So I when I get the courage up, I go swim with them. Um in the really cold water. It's freezing. Um and those people look like they're in great shape. So also correlation, but they're in their 60s with six packs. So um yeah, so I did a deep dive on the literature, and I have to say there haven't been a lot of studies. The studies are low quality, and there are not many of them. But from what I was seeing, there isn't much of a benefit to cold swimming beyond what you would just get from swimming. Um, and for muscle repair, so actually, in athletes that um, a lot of coaches were icing people's muscles after working out, because they were sore, and actually that impedes the muscle repair process. So I'm not actually sure that that is a good thing. So I came down on the side after reading. I came down on the side that's hot was good, but cold, maybe not.
Yeah,
Um,
yeah, yeah. Interesting. I mean, I remember hearing that for performance, for example, cold can be good, right? Because mitochondria, as they, get to, higher in terms of the glucose oxidation, right? They heat up, right? And then, there's a limit to the temperature before, proteins start denaturalizing or whatever, right? So if you're doing exercise very intensely, then take a break, go outside, sit in, minus zero temperature for a few minutes and then go back. You can keep performing at a pretty high level, right? But I haven't heard that it impedes I guess, the growth of the muscle or whatnot.
Yeah, I mean, you're reducing inflammation, and you actually need some of that inflammation to tear. Your muscles get torn, and then they repair, and that's part of how they get bigger, and more fit. So if you're impeding that process, you may be impeding the repair. Um, but that was just my read of literature. I wrote a Substack on it, so I've cited all the sources if people want to read my take on it. But
Yeah.
there really weren't very many papers published, so I think the jury is still out on it.
Yeah. Oh, yeah. I remember now somebody mentioned, I think at some point, yeah, like if you take a Tylenol or similar after working out, that is a very negative, right? Because you basically prevent the entire effect of the workout, right, from being beneficial to you. I guess for this like systemic reduction of inflammation.
Right. It's probably similar kind of principle with that. Yeah.
Cool. Maybe I'll ask one last question, and like we're already almost at the hour mark. Um, if if you could wish for one thing, that would make either, your work easier or the work of your company better, right? Or, one, technology innovation that you say, okay, it would be great to have something like that. If you had a magic wand, what would you wish for? You know,
For my company, um, I had a magic wand. I wish that venture capital was more efficient. So I think as entrepreneurs, we spend a lot of time fundraising. So I wish someone would come in and make that whole process better, match make people so you meet the right investor at the right time, and you know uh, there's some standardization to it, and because we waste a lot of time as CEOs fundraising. So that's one thing. Um, I think just better connectivity in general. We're partnered with 300 MRI clinics around the world. We'd like to be partnered with all of them. So there's some inefficiency there. I'd like to be able to send people for an MRI absolutely anywhere. So I think we need to, in the same way we have Uber and DoorDash and Google Maps, we need all of that in healthcare. So we need to be able to send people for anything, anywhere, anytime. So I think that would be on my bucket list of things, just improving the efficiency in that way.
it would be pretty amazing if you could have a $100 MRI, right, and then just do it, a couple of times a year, right? Same as all the rest of your blood tests or whatever, right?
Yeah, I wish it was in CVS, then I just walk there and just go get you know.
Mhm.
And I think people are starting to do that. I heard that I think his Target is starting to have mammograms, which is amazing. So you go shopping, then just get your mammogram while you're there. So I think it's coming, but that's what we need. We need healthcare to be way easier, more accessible.
Yeah, yeah. Well, I mean, an MRI would be even better, right? Because there's no radiation whatsoever, right?
Mhm.
Like a
Yep.
regular CT or X-ray or whatever.
Yeah, and the prices are coming down. So I think you can get a full body MRI machine for about $1 million now. 500K if you get one that's not premium. So,
Mhm.
you know, it's doable. I'm thinking about it. Should we just buy an MRI, like a roving MRI in a van? Um, I think
Yeah,
I think that's gonna happen.
yeah, yeah.
Yeah.
I'd say. Well, yeah. I mean, I would love to do that. It's like what's it called? The DEXA scan, right? You just, somebody puts the CT scan or the scan on a van. They drive around at a different location every day, right? So you could do your whole body MRI. Yeah. I mean, I don't think the MRI machine would work very well instead of a metallic van, right? But yeah, same idea.
Yep. Yeah.
Awesome. Cool. Well, this was fantastic. So thank you so much. It was a pleasure.
Yeah, thank you so much, Javier. I really, really had fun. Appreciate it.
Here. Thank you, Kristen.